Welcome to this lecture on fertilization, a fascinating topic in reproductive biology. Today, we are going to explore the very beginning of a new life. We will look at the incredible journey of the sperm, the biological mechanisms that allow it to fuse with the egg, and the mathematical realities of this high-stakes race. By the end of this session, you will understand not just how fertilization happens, but why it requires such astonishing numbers and precise timing. Let us dive in. To start, let us imagine a massive, high-stakes marathon. Millions of runners are navigating a treacherous, acidic obstacle course to reach a single, highly guarded fortress. This fortress represents the female egg, and the runners are the male sperm cells. Why does this analogy matter? Because it highlights the sheer difficulty of the journey. Only one runner is allowed inside this fortress. The exact moment that single runner crosses the threshold, the fortress doors lock permanently to keep everyone else out. This incredible race is the essence of fertilization, a story of endurance and precise timing. Now that we have an intuitive picture, let us define this formally. In biological terms, fertilization is the complex sequence of events where a haploid male gamete, known as the spermatozoon, fuses with a haploid female gamete, known as the oocyte. This fusion forms a single diploid cell called a zygote. It is crucial to understand that this is not just a simple collision between two cells. It is a highly regulated, sequential process. The genetic material from both parents combines here, transitioning from two half sets of chromosomes into one complete, brand new genetic blueprint. Building on that definition, let us look at the specific steps involved. First, the sperm must undergo capacitation. This is a biochemical maturation process in the female reproductive tract that makes the sperm hyperactive and capable of fertilizing the egg. Without capacitation, the sperm is essentially shooting blanks. Next, the sperm must physically push through the outer layer of the egg, called the corona radiata. Once it reaches the next layer, the zona pellucida, it binds to it. This binding triggers the acrosome reaction, releasing digestive enzymes that literally melt a path forward toward the egg's membrane. Following the acrosome reaction, we reach the critical moment of entry. The membranes of the successful sperm and the egg finally fuse. This fusion is the trigger for an immediate and dramatic defense mechanism called the cortical reaction. The egg releases cortical granules that rapidly harden its outer shell. Why is this so important? Because it creates the block to polyspermy. If multiple sperm were to enter, the zygote would have too many chromosomes and would not survive. This rapid hardening ensures that only one sperm contributes its genetic material, locking all the others out. Having discussed the mechanisms, let us now visualize them on screen. Here you see the large female egg on the right, surrounded by its protective layers. Notice the outermost cellular layer, the corona radiata, and the inner ring, the zona pellucida. On the left, several sperm are attempting to penetrate. Look closely at the sperm that has reached the zona pellucida. It is undergoing the acrosome reaction, releasing enzymes to break through. Just inside the egg's membrane, you can see the cortical granules. Once that first sperm fuses, these granules release outward, hardening the shell and creating the block to polyspermy you see labeled here. Now that we understand how it works, we must ask why this precise mechanism is so vital. Fertilization serves three absolutely critical biological purposes. First, it restores the normal diploid number of chromosomes by combining 23 from the sperm and 23 from the egg. Second, it determines the genetic sex of the new individual, depending on whether the successful sperm carries an X or a Y chromosome. Third, it activates the dormant egg to begin the rapid cell divisions that will form an embryo. Understanding this is also the foundation of modern reproductive medicine, including in vitro fertilization and contraceptives. Moving from biology to physics, let us consider the monumental physical task the sperm faces. The sperm must travel a specific, massive distance relative to its size, from the cervix all the way to the ampulla of the fallopian tube. We can actually model this journey using fundamental kinematics. By looking at the relationship between time, distance, and velocity, we gain a mathematical perspective on reproduction. This helps us understand exactly why millions of sperm are needed, and why a sperm's swimming speed is such a critical factor when doctors diagnose male fertility issues. This brings us to the mathematical formula that models the sperm's journey. The equation is T equals D divided by V. Let us break down what each symbol means. The variable T represents the total time required for the sperm to reach the egg, usually measured in minutes. The variable D is the total distance through the reproductive tract, measured in millimeters. Finally, V is the average swimming velocity of the sperm. Physically, if the distance D increases, or if the sperm's velocity V decreases due to poor health, the time T increases significantly, drastically reducing the chances of success. Before we move on, let us see this formula in action with a clinical example. Suppose we are evaluating a patient. We know the total distance the sperm must travel is 150 millimeters. The average swimming velocity of this patient's sperm is 3 millimeters per minute. We want to find how long the journey takes. We choose our kinematic equation, T equals D over V, because we have distance and constant speed. We substitute 150 for D and 3 for V. Dividing these gives us exactly 50 minutes. This means it takes 50 minutes of continuous, vigorous swimming just to reach the egg. Now that we have calculated the time required, we must face a harsh biological reality. The journey is incredibly dangerous. The probability of any single sperm surviving the acidic environment, navigating the cervix, and successfully penetrating the egg is incredibly small. This is exactly why a healthy ejaculate must contain hundreds of millions of sperm. To understand this mathematically, we can use probability theory to calculate the overall chance that at least one sperm out of the entire population will successfully fertilize the egg. To calculate those odds, we use this probability model. Capital P equals 1 minus the quantity 1 minus lowercase p raised to the power of n. Let us define these terms. Capital P is the overall probability of successful fertilization. Lowercase p is the tiny probability that any single individual sperm succeeds. Capital N is the total number of sperm attempting the journey. Conceptually, as the total number of sperm N increases, the overall probability of success P gets pushed much closer to 100%, ensuring the survival of the species. Let us apply this formula to a specific case. We are given a very low single sperm success rate of 0.00001 and a low sperm count of 50,000. We want to find the overall probability of pregnancy. We use our probability model and substitute our values.
1 minus our tiny probability gives a failure rate of 0.99999 per sperm. We raise this to the power of 50,000, which gives approximately 0.6065. Subtracting this from 1 leaves us with 0.3935. Physically, this means there's only a 39.35% chance of successful fertilization for this cycle.
Now that we have used this formula, let us understand where it comes from logically. We want to find the probability of at least one success. Mathematically, calculating at least one directly is very difficult. Instead, it is much easier to calculate the probability of 0 successes, or total failure, and subtract that from 1. If the chance of one sperm succeeding is p, then its chance of failing is 1 minus p. Since all n sperm are swimming independently, the chance that absolutely every single one of them fails is 1 minus p raised to the power of n. Therefore, the probability of at least one success is simply 1 minus that total failure rate.
To wrap up our lecture, let us review the most important ideas. First, fertilization is the highly coordinated fusion of haploid gametes to create a genetically unique diploid zygote. Second, mechanisms like the block to polyspermy are essential to ensure exactly one sperm fertilizes the egg, maintaining the correct chromosome count. Third, the physical journey of the sperm is governed by simple kinematics, where time depends on distance and velocity. Finally, because the individual chance of success is so microscopic, millions of sperm are absolutely necessary to guarantee the continuation of human life. You now understand both the biology and the math behind the beginning of life.